Today we will receive an overview of the up-to-date virology and immunology issues involved with the current coronavirus pandemic and how Biochrom can assist supporting those customers who are doing research and antibody testing. It is my pleasure to introduce Rachael Nugent. Rachael serves as the Global Marketing Director supporting the molecular portfolio at Harvard Bioscience. Graduating from Queensland University and majoring in microbiology and biotechnology, Rachael jumped into the hospital setting to gain practical experience by working in a clinical pathology laboratory, focusing on tissue culture and serology diagnostic approaches. From science to sales, Rachael transitioned to become a capital equipment specialist for the UK's largest scientific equipment distributor. And in 2014, she joined Harvard Bioscience as a distribution sales manager and recently transitioned to the marketing side of the organization earlier this year. Rachael, thank you for being with us today.
Thank you very much, Mike, for the great intro. So to get stuck into it, viruses are, I think, beautiful, but that's possibly just me. So all viruses, they have different sort of strange surfaces. These surfaces, they're little spikes, the bubbles, the different patterns that you see on them. These are the antigens. These antigens are basically proteins 99% of the time. And the antigens are what allow the virus to attach to the cell of the host. They attach to the walls of the cells, and that's how they penetrate through the cell membrane. Once they're inside the cell, they tend to use the cell as sort of an incubator. They take the nutrients, they take the warmth, they take the fluids to feed themselves. They then multiply and multiply and multiply, swelling up that cell and then bursting out to then go and inhabit other cells. Ideally, in a good best case scenario, the body, the host, will have an immune response. And how that works is the body will produce antibodies to those antigens, and that antibody-antigen interaction is what all of this sort of really relies on. And the antibodies, sometimes called neutralizing antibodies, sort of bind against those antigens and sort of coat them in a way that stops them then being able to adhere to the cell walls in the first place, and then stops that entire process from the entry to the reproduction. That's also very much in an ideal situation. That's the typical progress that we see. It doesn't happen in every virus. And as you can imagine, there are certain things we don't know about the coronavirus. Coronaviruses are not new. Coronaviruses have been sort of identified in literature since sort of the early 1900s, but we expect they've been around for a lot longer than that. The first sort of real identification and the naming of it actually happened in the 1960s. So the current one we're looking at, it's a coronavirus. Yes, that's the family of viruses it belongs to. We can call it COVID-19. We can call it SARS-CoV-2, that to distinguish it from SARS-CoV-1, which was the initial SARS outbreak in Asia in about 2003 that some of you will remember. So this coronavirus, all coronaviruses are RNA-based as opposed to DNA-based, and obviously they attack the respiratory system primarily. This coronavirus, the COVID-19, it has sort of three main surface antigens, remembering those are what allow it to actually adhere to the cells and penetrate the host. You can see from that picture, which is lovely, the spike protein, which is the S protein there, and those are literally the spiky bits. That's why it's called that. It's because they look spiky. The envelope protein, which is the E protein, and the M protein, which is the membrane. Now, these are sort of the three primary surface antigens that are around the COVID-19 virus and what allow it to actually infect cells, whether they be human or bat or whatever. Now, antibody testing, which is what we're going to be talking about today, requires that the host creates antibodies to one or all or some of these proteins, these antigens. Now, each of these have pros and cons. Some of them may provide protection, some of them may not. It's something that we're still really working on from a scientific perspective. But this antibody testing that we're going to be talking about, usually it can be done on a variety of different samples, can be done on urine, it can be done on saliva, depending on the virus, to be honest. So not all of them can be done that way. We're looking at the coronavirus and COVID-19 for antibody testing primarily through a blood test, in which case you separate the blood out. So you're looking at the actual serum of the blood, so it's removing the red blood cells. Now, a lot of the news, obviously, we're spending so much time listening to it, people talking about testing, and you're seeing images of the nasal swabs and things like that. There are two tests that basically we're talking about when we're referring to COVID-19 and the current issue. The first is the testing for active infections. So if I were to get sick, that would be what you're looking at there, would be the timeline of how we would actually go about testing. The total of that is about sort of 20 to 22 days, and that's that length of that axis there. So the viral RNA, that nasal pharyngeal swab that you're seeing with the swabs going up the nose, that is actually a PCR test, and that is to detect active virus. So you can only really detect it in that sort of, say, 5 to 9 day period right at the beginning and right at the onset. That you really need to be swabbing and finding virus particles in the nose to actually be detecting it. As that drops off, and this is a pattern that you see with most virus infections, to be honest, you get the body's response. So, and that's the IgM and the IgG that you can see there. The IgM is the neutralizing antibodies that you get an initial production of, and that is what dispels that first infection to an extent. And the IgG is, in theory and hopefully, what your body can produce once it's sort of adapted, once it's gotten itself together. And hopefully the IgG antibodies are what's going to give you some form of immunity. Now, one of the strange things, one of the little quirks about COVID-19 is that big gap between that blue box and that pink box there. Normally you would see those a lot closer together, and you would see a lot more crossover with regards to those two types of tests being relevant and being able to actually give you an answer. So we do see a gap in that, which is strange. I'm sure somebody's going to tell us why that's a thing at some point, but we don't really understand that yet. So with regards to antibody testing, there's a lot of talk about these point-of-care tests, sort of pregnancy-type tests, that in theory you could prick your finger, put a little bit of blood into the port at the beginning, and it's a lateral flow antibody test, in which case that little, so it's a sort of a filter paper type situation between two bits of plastic, and the blood will flow along there, and it will pick up antibodies at the same time, and then it will have a color reaction at the end of it. Now, that in theory is great and would be really helpful in this situation. No one's been able to actually produce one that is sensitive enough yet. We've had a lot of reports about, I know the UK government bought something like 3 million of these tests and none of them were effective. So we're not there yet. Ideally, I think that people would like to be seeing those so they can test themselves regularly, so they can say, well, look, I'm still producing antibodies to this, so I know that I have immunity. But as we said, one of the potential things is we don't know how long that immunity is going to last for. We don't know whether the presence of antibodies is actually a good indicator of immunity, and a strong immunity at that with regards to coronavirus at the moment. So while this is the goal, and that would be something that you could probably buy in your local pharmacy or drugstore, we're not there yet. I don't know that we're particularly close, to be honest. What we can do is the lab-based antibody test. Now, the most common one of these is an ELISA test, or enzyme-linked immunosorbent assay, which is a bit of a mouthful. ELISA tests have been around for a long time. I think the first ones were actually sort of brought to market in the 60s. So they're generally purchased as a kit. You need to have a blood test, so you need to have blood drawn. That blood needs to be processed in a lab, and it does use quite a bit of equipment and lab resources to do that. They're usually processed in a 96-well plate for, throughput. And that's generally the setup you would see in a 96-well plate there. You really need to do a standard curve with this, so you need to actually have known samples that can give you quantitation, because it is a quantitative assay. We want to know how much antibody you have, because hopefully there is a comparison between the amount of antibody you have, and the more you have, the stronger the immunity that you have to that virus. You also need positive and negative controls. So all of these are done also in duplicate. So you can see the first two columns there, the second two columns. That's how you would actually put the test. So the patient samples would be in the number 11 and 22. So that's what it would generally look like. It's typically in a 96-well format. And these tests generally are purchased as a kit. So what you actually buy is a kit, a box that has a plate, it has the reagents you need. That plate is in a sealed packet, and the wells of the plate are actually going to be coated with an antigen. And so they're very specific to the virus that you're looking for. So in the theory of this, buying in an ELISA test for COVID-19, you would have, say, the spike antigen coating the bottom of that well of the plate. There are companies that make antigens just separately to be developing these types of assays as well. And not all antigens are made the same and made equal. So once you've got that plate out, you've got your kits, you've got your reagents, you need to put in some serum, patient serum. There are a few steps to this. There are also a couple of different types of ELISAs with varying numbers of steps. But this is just an example of a direct ELISA. So you've got, if you imagine each of those three black lines as the well, you start off with your virus sample, which is your antigen that's bound to the surface of the plate. You would then add in your patient serum sample, and then you'd leave it for a little while. And what happens if there are antibodies present in that sample, they're going to bind to those antigens. So once they've bound, you actually need to then wash off all of the excess. So there's going to be lots of serum and other bits and pieces. Because it's a quantitative assay, you don't want any extra bits and bobs in there. You would then add on something, a substrate or conjugate, to attach to that antibody. And what that will then show is a color reaction. Again, you're going to need to do a wash step in there. But that color reaction, usually, to my mind, my experience, they tend to always be sort of yellow or blue. You can actually see them visually, and you can see the variation in them. So you can see, well, okay, that one had a really strong reaction, so it must have a lot of antibodies. That one, oh, there's a bit of color change, but not much, and that one doesn't have any at all. To actually, obviously, put a number to this and to quantitate it, you would want to do that not by eye, but via an instrument. So at which point you would put it into a microplate reader. And usually, like I said, these are, very, very, very simple instruments. The ELISA readers do not need to be complex. And that will give you a level of absorbance. So the higher the intensity, the higher the absorbance. And that absorbance reading will be able to quantify against that standard curve that you've entered in your plate earlier. The EasyRead 400 ELISA. So there are two versions. There's the ELISA and then there's the research model. You've got the part number there. It's a pretty basic instrument. It's got filters that drop in and out and can read at various wavelengths. It is a visible only instrument. It is an absorbance instrument. So the instrument itself is supplied with those filters there, the 400, the 450, the 492, and the 620. We've been keeping an eye on all of the various kits that are coming into the market to be testing for COVID-19. The majority of them seem to be reading at 450, and that's the active read point. So while it is possible, because these are still in development, each country and within each country, each lab even, or each government organization is developing their own assays, and how they develop that is going to be very much reliant on what substrates they have access to and what supply chains that they have no problem with. So we don't see it at this point going much outside of those wavelengths. However, there is potential for that to happen. Now, that 620 wavelength is often used as a reference, so you can sort of be blanking on it and things like that. But none of the ones that I've seen are actually talking about reference wavelengths. So like I said, that 450 is the one that seems to be the primary option. The instruments themselves are supplied with the software that's required to run them. The software, whether you can see that screen on that laptop in the picture, basically gives you a plate layout like you saw earlier, and you can just drag and drop drag and hold across the plate to set up your assays and things like that. It is a Windows-based software, so just make sure that they're not running on Macs and things like that. And it does come with the cables to attach up to a USB. One side of the cable obviously goes into the instrument, and the other one is just a basic USB plug, but we do not supply the PC, so the customer will need to use their own PC for this. So that's what comes as part of the package.